So in a slight change to the advertised programme, we're going to do the panel discussion on omics now. So could the speakers all please come up? And I'd also like to invite Kathy Sudlow as the Chief Scientist of UK Biobank. Kathy's worked very hard already today. I'll ask her up one more time. So, you know, there's quite a challenge been thrown down there. I was keeping track of the numbers as the speakers were delivering their presentations, and the biggest one I heard was 20,000, and now we're talking about 500,000. I'm sure there will be comments and questions on this. This is very important. UK Biobank is definitely looking for your input. So do we have any questions or comments from the floor? Okay, well, I'll take chair's privilege. Kathy, I'll ask you. From the UK Biobank perspective, what's your thoughts on what you've heard, and in particular with regards to the scalability of some of the technologies that have been talked about?
Yeah, thanks, Bernard. So I'm not an omics expert, so all I'm going to do is state the blindingly obvious, which is to say that it's very clear from what all the speakers have told us that the size of UK Biobank and its existing rich, diverse data make it an obvious place to start this scalable proteomics and metabolomics experiment, and to add that additional richness for the power of combining additional data with all the data that already exists to increase understanding of disease mechanisms, to improve the power of current predictive tools, and move towards novel biomarkers and novel diagnostics, new therapeutic targets, and improve the power of subphenotyping and discovery of new disease subphenotypes. And I would just add that because Biobank is so widely used and widely available, and because this will undoubtedly raise new, quite substantial scientific challenges, the fact that Biobank is being accessed by so many scientists will mean that this initiative will be transferred much more quickly into usable knowledge by the use of many minds all over the world.
I think that's a critical point you make there. Thank you. Actually, if we could pass the microphone along to the other speakers, maybe they would like to make comments on that same point.
Is this on? Yeah, so I think I tried to make the point in my last slide, really, that one of our challenges right now is just statistical power. You know, I think we had done a study of incident pancreatic cancer a few years ago, found indicators that predicted diagnosis of pancreatic cancer up to 6 years in advance. But those people probably already had pancreatic cancer, it was just subclinical. But we had to reach out to 4 different cohorts and cobble together a bunch of samples, where I think in the end we ended up getting something like 500 cases or 400 cases. But I think I saw in some data that you already have something like 4,000 plus incident cases of pancreatic cancer in the biobank. So it's sort of maybe, I thought about it maybe career ending to do the study, but I think it's pretty compelling.
So yeah, my comment would be from our experience in the interval study about scalability. As you've seen, we used the SomaLogic platform and the Olink platform essentially because they were at the time more easily scalable. I think mass spec, as we heard from Tony, is kind of now getting there. I think when we embarked on our work, it kind of wasn't, and that's why we went for the more targeted assays. But I think now is probably the time to go for mass spec.
Yeah, all right, is this on? Yeah. I mean, I think the thing, what really struck me most to work with these things is that you get really annotation of this mass spec peak, so that you get really an idea, not a study of peak 1 to peak 11, but to have a direct annotation of all the metabolites and to link them to pathways and then to the disease. And I think then you can also argue more like in a Bayesian way to say like this metabolite really makes sense in this context. And you also get a feeling, very often you get a feeling for whether something is a true positive or not. Right, so having this annotation, I think, is really the key thing.
Thank you. Tony will let you off because you issued the challenge, so we'll let you off this one, and we'll go to number 3 at the back.
Hi, Roberto Bonelli, Australia. I want to ask the panel, since, by the way, thanks so much for your talks. Since we're moving more and more towards the disease now, so we start like from the genetics, we're moving closer to the disease, and we expected, especially in terms of statistical signal, to have stronger and stronger signal. What I wanted to ask your opinion on is, do you think it would be better if we, as you said now, like we start doing proteomics and metabolomics on the entire cohort, or if we say set a golden subcohort, and on this golden subcohort we start actually doing all the levels between genetics and disease, so also including RNA-seq, epigenomics, microbiome, and such.
Okay.
Shall I answer that? I think the danger is that you introduce a batch effect that's confounded with what later turns out to be a variable of interest, even if at the time you try and design it in a way that it doesn't. You later want to look at some other variable, and you've induced this sort of artificial technical effect through the cohort. So I think really you want to randomize your samples as much as possible and probably really do the whole lot.
Yeah, and maybe if I can add to this, I think also the point is that UK Biobank is really to have 500,000 people and having all this kind of being able not just to say, okay, there are 2,000 cases, but we have all these other variables we can stratify. I mean, smaller studies, we've seen a lot of them, but I would definitely say the strength of UK Biobank is to have 500,000. Going beyond 500,000, I wouldn't compromise on that.
Great. Question number 2 then.
Can I just remind colleagues, right, about 20 years ago, there was a major effort in using mass spectrometry to identify biomarkers in fluids, right? And the NIH had a project called Early Discovery Research Network. And we spent more than to the order of $1 billion to try to identify biomarkers in fluids and plasma and all that for chronic diseases like Alzheimer's disease and as well as for cancers, right? And it was a massive effort, and we didn't really succeed. Basically, there were technological challenges, there were scientific challenges and all that. Now, what you outlined here is important. I agree with you that the science can be addressed. The technological challenges, we have moved much further than we were 20 years ago, right? But there's a fundamental problem that you need to consider, which is the cost of generating this data, the cost of maintaining this data and sustaining and growing it is incredibly expensive. And we need to think about a business model that will actually sustain this whole process, right? If we don't take that into account, it's going to be, you will start off with something and then you'll be caught in a certain trap. And you mentioned in one of the talks Lee Hood's effort to do this comprehensive study on 128 people, right? It was initially funded, they spun out a company. And I think that the company is defunct now because they couldn't afford to sustain and grow that population to 1,000 or 10,000 potentially they wanted to do.
Okay, so your question then is to do with cost and sustainability. Yes. So, Tony, would you like to?
So the first thing to say is that Danaher SciEx have indicated that they would be prepared to put funding into this project. But I want to go back to the first point you made. So the investment around biomarkers, etc., was in, I think you're referring to the use of cell-detoff technology, which in more or less every case, you didn't know what the entity was that you were measuring. So mass spec, I agree that the cell-detoff business and the work that was done around ovarian cancer biomarkers set the field back a very long way in terms of proteomics and clinical proteomics. So I'm agreeing with you on that, but as a user and somebody who's watched this field, I think that the technology has developed to an extent where we have the degree of specificity around what we're measuring to the degree where we're in a good place. So the big issue with UK Biobank and measuring proteins is going to be about the specificity of what you measure. And Jimmy referred to this, he referred to the issue of sensitivity. What is binding? With mass spectrometry, you have structures and you have information which enables you to actually identify the chemical nature of the entity you're measuring, both in terms of peptides and therefore proteins. So I'm comfortable that the field has advanced away from that cell-detoff era.
Okay, so we'll take one, which will have to be the final question, I think, at the back.
Well, a question to all the panel, but particularly the scientific officer for UK Biobank. How are you going to choose between different competing consortia for these kind of measurements and for these kind of studies? There are at the moment a range of different platforms. Every single lab will give you a different result. And I know Dr. Klisch showed some really impressive data, merging different data sets over long periods of time from different cohorts. But at least in the metabolomics community, less clear in proteomics, there's precious little data showing that you can get agreement between laboratories using these kind of untargeted methods. So to what degree are these kind of factors going to be a criteria for selection? Or how are you going to choose between what are going to be a myriad of different consortia offering potentially very different solutions?
Yeah, so it's a very relevant question. I thought you were going to ask me about how we choose between which researchers would be able to access the data, to which the answer is all of them, of course. But actually, because your question is much more about the selection of appropriate platforms to run assays, I'm going to bounce it over to Tony.
Well, I'll talk about proteomics, and with the kind of data-independent acquisition / SWOT-MS type approach, then the information that we have on reproducibility between different laboratories is good. It's a kind of platform with the use of reference standards that enables you to perform these kinds of major studies. I think I should let metabolomics people answer.
Well, the data I showed was all from our lab, but in my opinion, I think it makes sense that it's probably a good idea if you choose a particular method. I showed that we had 4 that we might use, but it's probably good to run that one method in one place across the whole study. But it doesn't mean that you can't have multiple labs contributing, because other labs could be running different methods.
So maybe if I can just add to that as well. I mean, we have also the example of the Nightingale project already. So I think there is certainly also an incentive not at all to have kind of UK Biobank fund this, but rather to have a model like that and to say, look, that was the last slide that I showed where you see all these cohorts. You could partition that and say, okay, everyone interested in the phenotype could put money in there and then go for a project like that.
Yeah, so I mean, so that really just picks up on the last point I wanted to add, is clearly this depends on costs, and there will be some sustainable cost models. There'll be some potentially interested industry partners who might be very interested in helping to fund this kind of effort. And so, coalitions of partners may come together to help with funding preferred platforms. And so it's going to be a combination of scientifically excellent platforms, perhaps some additional studies to look at quality with particular reference to the biobank samples, scalability, and cost.
Right, so we'll leave the omics discussion there. Thank you very much to all of the panel.